This is the methodology that we applied. We used the chip arrays from Affymetrix. We did, obviously, this process started from the histological assessment, and the tumor cells and the tumor that we used were only the tumors that had the content of at least 80% of tumor cells. We verified the diagnosis and the grading, and then from those specimens we extract RNA. We did the quality control. We processed the RNA according to the Affymetrix protocols, and we did the prioritization of the chips. The statistical analysis was done by Siemens and by Alessandro's group. The statistical analysis, as I said, was done by Siemens, and they did, this is all the statistical analysis that Siemens did using the normalization, and then they applied t-test, student t-test analysis, and then the integration of Siemens and Dizzy analysis that Alessandro will present us will give us all the genes at the end that Samantha will talk about later. So the method, the microarray analysis was done of pilocytic astrocytoma, as I said, for biological characterization. And this is the heat map, the first heat map on pilocytic astrocytoma, and the first, as you can see, we were able to see all the information that came from this heat map, and we were able to find all the, as you see, the red and the blue, and we were able to distinguish pilocytic astrocytomas, and we were able to distinguish the group of genes that differently expressed from cerebrum versus cerebellum. And then the next group of tumors, pilocytic astrocytoma, was cerebrum versus midline, and we were able to identify another group of genes. And as you can see, we were able to distinguish other list of smaller genes, and were able to distinguish the cerebrum versus midline. And then between always in pilocytic astrocytoma, we identified other genes that were specific from cerebellum versus midline. And then this was the first group of tumors that we were able to study. And then our next analysis was just on all low-grade glioma, means pilocytics plus other histological types, and we wanted to understand the genetic characterization upon site of lesion. These are all the, so it means pilocytic astrocytomas versus ganglioma, and this heat map again. So microarrays, the technique of microarrays is able to give us information of 47,000 different genes, and at the end, the statistical analysis and the integration of the statistical analysis from microarray and confirming those data from using the quantitative PCR will give us lower number of genes that we are at the end able to understand at least part of the genetic components of this kind of tumor. So it's a long process coming from a huge group of expression studies, a group of genes. At the end, we will able probably dealing of analyzing 22 genes using quantitative PCR, and now Samantha will present us.